Hi, Michelle Glass here. Welcome to our first lecture in our Chapter 8, Lecture 8 series. Our topic for Chapter 8 is all about cell division. And the significant part of that is that transfer of DNA material from one cell to the next. So we need to take a few minutes and talk a little bit more about that molecule. So we've talked about it before, and we're going to talk about it here, and we're going to talk about it again in the future. DNA is, of course, standing for deoxyribose nucleic acid. It is our molecule of inheritance. Now, all of the DNA in the cell is referred to as the genome, and it's made up of units called genes, and each gene is coding for a protein. When we talk about the cell preparing for division, one of the things it does as part of that is to package up that DNA into a structure called a chromosome. And this is basically just wrapping that DNA around proteins. The significance of this is we don't want to get it lost or damaged in the process of moving. So just like you box up all your worldly possessions when you move from one apartment to another, in the case here, the cell is going to pack up all of its DNA into this condensed form, wrapping it around the proteins, and we call that a chromosome. So we only see chromosomes in the cells that are dividing. Now, when we talk about chromosome number, we'll see that that number is species specific. So there is a lot of variation here. Humans have 46 total chromosomes in our cells. Elephants have 56. And cabbage has just 18. Well, one of the things that we will see as a common theme is that these numbers are always going to be even numbers. And the reason is because half of that DNA material, half of your chromosomes, are being inherited from the egg cell coming from the mother, and the other half of the chromosomes are coming from the sperm cell donated by the father. And so we see that our chromosomes are always paired because of this relationship. And what we'll see is that they actually will match up. So you'll have two number ones and two number twos and two number threes and so forth all the way as we look at our total number of chromosomes. When we're talking about a cell that has paired chromosomes, meaning two number ones, two number twos, and so forth, we're going to call that cell diploid cell. And we can abbreviate that as 2N. So a human cell that has 46 chromosomes, those are paired, so that's the diploid number. The elephant cell with 56 chromosomes, those are paired, so that is the diploid number. We will see for animals especially, and for most of the plants, that our somatic cells, or the cells of our body, are going to be diploid. What we're going to typically see is that the gametes, the sex cells, the egg cell, and the sperm cell are going to be haploid. And so haploid here means half of a set. So in the case of 46, the egg cell contains 23, and the sperm cell contains 23. This is true for cabbages also. So the egg cell is going to contain 9 chromosomes, and the sperm cell, which is in the pollen, is going to contain 9 chromosomes, so that when that pollen grain fertilizes that egg cell, we get a developing embryo or seed that will have 18 chromosomes. We've talked about chromosomes as that condensed form of the DNA molecule, wrapped around the proteins, ready for division. Okay, but the rest of the time, a chromosome is described as being in its relaxed state. So it's all spread out, watching TV, whatever, studying biology, and we call that form of DNA the chromatin. When we talk about DNA replication, and when we talk about gene expression, or the process of converting a gene into a protein, or taking that gene sequence, maybe I should say, and using the sequence to make the protein, that's going to be in the chromatin form. The chromatin relaxed DNA is going to be found when the cell is either just hanging out doing its job or getting ready for division. And we call that phase interphase. And we're going to have a whole conversation about interphase in the next video. So we'll talk some more details. Most of the time, the cell's in interphase, so most of the time, the DNA is going to be in that chromatin form. When we talk about cell division, we need to take one set of DNA and then double it and then make 2 new cells. And so it's important to pay attention to when that process of doubling the DNA or replication occurs. DNA replication is going to happen during that interphase when the cells are relaxed in the chromatin phase. We will see that you will have what's called a chromatid, and it will duplicate itself so that you will have from one chromatid to 2 chromatids, and we will call these 2 chromatids sister chromatids. So this is a specific term. Sister chromatids are attached at a location called the centromere. So we go from one chromatid to 2 identical twin sister chromatids attached together at the centromere. And we'll come back to this idea in the future. So make sure that you're paying attention here and looking at this. The other thing that's interesting is we count our chromosomes by our centromere numbers. So we have 1 chromatid and 2 chromatids. Let me rephrase. We have 1 chromatid on the left side of the arrow, 2 chromatids on the right side of the arrow, but in each case, we just have 1 chromosome. Okay, that's it for now.